Welcome to our educational webinar entitled Brain Organoids, Modelling Human Brain Development in a Dish. My name is Sam Lloyd-Burton and I'm a Senior Product Marketing Manager at STEMCELL Technologies. It's now my pleasure to introduce you to our speaker today, Dr. Madeline Lancaster, whose groundbreaking work in the development of cerebral organoid technology is revolutionizing neurological development and disease modeling, previously worked in the lab of Dr. Jürgen Knoblich. She followed through on an unexpected observation that she would later develop into three-dimensional cerebral organoid structures, colloquially referred to as mini-brains. Today, she leads her own research group at the MRC Laboratory of Molecular Biology in Cambridge, UK. Madeline's research focuses on studying neurodevelopmental disorders such as microcephaly, autism, and intellectual disability, and examining the roles of mutations in pathogenesis in the context of cerebral organoid development. She also studies cellular mechanisms underlying neurodevelopmental disease progression and potential therapeutic avenues, and is interested in performing comparative evolutionary studies to identify conserved or unique factors in human brain size evolution. Now let me hand you over to Madeline to begin the presentation.
So thank you very much for this opportunity to give this webinar. It's really a pleasure to be able to tell you a little bit about brain organoids, what they are, and what they're capable of modeling. So I'll start by just sort of giving you a background on human brain development. The human brain is probably one of the most intriguing organs in the animal kingdom, and I think it's because it's mainly what makes us unique as a species, because it gives us these unique abilities like logic and reasoning, speech, complex emotions, and social interactions. And so understanding where those complex capabilities come from and how our brain gives us these abilities is something that I think neurobiologists for hundreds of years have really been intrigued by, and something that I'm also very much interested in. Much of what we know about the human brain actually comes, of course, from model systems like mouse. Because of many ethical and technical limitations, it's been very difficult to really study the human brain. And so the mouse brain and also other model organisms have really given us a lot of insight into mammalian brain development and function. But actually, if you look at other mammalian brains, what you find is that there's really quite a lot of diversity out there. And the mouse brain is a bit odd, actually, compared to the rest of the brains. And so which features of our brains are sort of specific to humans versus maybe just different from mouse is not really known. And there's just so much still to be learned about which features are specific to humans and which features really give us those important cognitive capabilities. But one thing is sure, that most of what gives us our unique abilities probably arises during development. And so I'm just going to sort of give a little crash course on brain development. Typically, when we look at the developing brain, we look at sections. So on the upper left here, what you see is just a drawing of a human fetus, and you can see the central nervous system here. And if you were to cut a section through the front of the brain, that's the cerebral cortex, the sort of dorsal part of the forebrain, what you would see is something that looks like this section on the bottom left, where you see this nice fluid-filled spaces here and the cerebral wall. And if you zoom in on the wall of the cortex here, then I've drawn this schematically on the right. And what you see is that you have this kind of layered structure of the cells. So you have the neural stem cells on the inside next to the ventricle. And these are also called radioglia. And they have several important characteristics. Because they're actually epithelial cells, they span the entire wall from the ventricular surface all the way to the outer surface. And they divide actually next to the ventricular surface. And when they divide, they can either divide and generate two more radioglial neural stem cells, and this would be a symmetric expansive division. And so if they do this type of symmetric expansive division, then they'll simply increase their numbers. But they can also undergo asymmetric divisions, where one of the daughter cells remains a radioglial neural stem cell, but the other daughter cell becomes more differentiated. And when it becomes more differentiated, it sort of moves outward. And these more differentiated cell types can be neurons directly. These are drawn here in blue. The neurons then use the radioglial basal process, this long process that connects all the way to the outer surface of the brain. And they use this process to migrate and find their position in the outer region, which is called the cortical plates, here, the CP. But other more sort of differentiated cell types that are sort of in between radioglial neural stem cells and neurons are these kind of intermediate progenitor types. These can be TBR2 positive intermediate progenitors, which I've drawn here in green, but they can also be other, there's a whole myriad of different types of sort of intermediate populations here, which also can include these cells called basal radioglia, which have recently been identified. And these cells are very much like the radioglial neural stem cells, except that they lack the apical connection to the ventricular surface. And so their cell bodies are somewhat displaced in a sort of an extra region called the outer subventricular zone, the OSVZ, written here on the left. Now, in terms of how this relates to human brain development, well, it's thought that there are a few places along this lineage where humans may show differences compared with mice that may be responsible for our increased neuron numbers and therefore increased size. One of which is this early divisions of the radioglial neural stem cells. These divisions may undergo more self-renewing expansive divisions before switching to neurogenesis, and that would be a way of increasing the founder stem cell population, and one could imagine that that would then lead to a larger number of neurons in the end. And another mechanism is by increasing these intermediate populations, like these intermediate progenitors and basal radioglia. By increasing these intermediate populations, those cells themselves can divide further and expand, and then also give rise to neurons themselves. And in that way, they could magnify the neuron number. And there's evidence for that at play in human evolution, because when you look at primate developing cerebral cortex compared with a rodent, what you find is that you have this ventricular zone with the cell bodies of the radioglia here, and you can see that in both of these sections here. And then the neurons that migrate outward and populate the cortical plate, the CP here. But in primates, there's this very dense OSVZ, this outer subventricular zone, and that is that region that is populated by those intermediate populations. And as you can see, that region is completely absent in rodents. Now, rodents do actually have some of these various cell types. It's not that humans have a cell type that doesn't exist in rodents, but clearly you can see it's very dramatically expanded in humans and other primates. And so it's thought that by increasing these various progenitor types, that would increase neuron number. But actually, a lot of this is really not well known. So these are all essentially theories, and there's not very much actual functional data to support this because of the both ethical and technical limitations. It's obviously difficult to do experiments with human material, and until recently, other types of in vitro models haven't really been available. And so a number of approaches have been taken to try to overcome this issue. And so my work, and in general the field of brain organoids, really builds upon decades of research trying to recapitulate these neurodevelopmental processes in a dish. And so the first description really of neural stem cells was back in 1989. And then in 2001 were the first neural rosettes. And these were really remarkable because they mimic the kind of epithelial morphology that neural stem cells display. And that's also important for their lineage then later. And then through some really seminal work in the lab of Yoshiki Sasai, who I think was really a founder of this field, they established this method called SFEBQ, which is kind of, we like to kind of call it 2.5D. So they started with 3D tissues and then sort of plated them down and ended up with these really complex tissues that are sort of somewhere between rosettes and organoids. And then I think the first real neural organoid was also done in the lab of Yoshiki Sasai, and that was these optic cup organoids. So these were these entirely 3D tissues. They didn't plate them down or anything, and they displayed this really remarkable self-organizing capacity to form essentially the neural retina. We then built upon this work and sort of took away any kind of growth factors or any kind of small molecules, and essentially allowed the neural tissue to develop itself. And we ended up with what we at the time called cerebral organoids. We sometimes also call them brain organoids, or colloquially mini brains. And I'm going to tell you obviously more about that. And then since then, a number of other methods have come out. I'll also tell you a little bit more about those. Here you can see I've also labeled the cortical spheroids, and I'll talk more about those as well. So basically what all of these different neural organoid methods take advantage of is the fact that stem cells can follow the same kind of developmental programs that you would normally see in a developing embryo. So in vivo, you get fertilization of an egg by the sperm, and then this starts developing into a blastocyst, the very, very early embryo. And the inner cell mass of this blastocyst is equivalent to the pluripotent stem cells that we use in the lab. And then this inner cell mass is what will give rise to the entire body of the embryo. So it undergoes some initial important morphogenetic events, giving rise to the germ layers that will eventually build the various organs. One of those germ layers is the ectoderm, and that part of the ectoderm will develop into the neuroectoderm, which is the precursor for the entire CNS. And that neuroectoderm will form something called the neural plate, which will then fold up and form the neural tube. And as you can see now, you can start kind of recognizing how that neural tube eventually develops into the brain of an embryo. And the most anterior portion of that neural tube is what will give rise to the brain and forebrain and the cerebral cortex. So the cerebral organoids basically take advantage of this and sort of essentially just sort of cultivate these cells and these tissues and allow them to develop and kind of do what they're already programmed to do. So like I said, pluripotent stem cells are essentially equivalent to these inner cell mass cells, and they can be lifted off the plate and then grown in these 3D spheroids, which start to spontaneously differentiate and try to generate the germ layers of an embryo. And these are called embryoid bodies because they're kind of trying to generate an embryo, but it's obviously a bit disorganized. So you have sort of different germ layers beginning to develop in various kind of random locations within the embryo. But then by placing these embryoid bodies in a media that is sort of limiting to the other germ layers and only allows the neuroectoderm to develop further, we can kind of purify these tissues to become more neuroectoderm and then drive them, well, and then actually they sort of self-organize and start forming these really nice neural tube tissues once we embed them in matrigel, which is basically a gel of extracellular matrix proteins that stimulates this neuroectoderm to reorganize and to form these buds. And each of these little buds then will display the same kind of architecture as the neural tube that you would see in vivo. So they have these nice fluid-filled cavities and with epithelial morphology. This is just kind of an overview of the method that we developed. I should say that this work was, of course, a collaborative effort, something that I started working on in Jürgen Knoblig's lab, and we really established this method together, and then, of course, worked with others to improve it, and I'll talk about some of those improvements in a little bit. But basically, this was the initial protocol that we published, where we start with the generation of these embryoid bodies, and then through a series of just sort of media changes, moving them through different types of media, as well as embedding in these matrigel droplets, we can finally get these really quite complex tissues, which then can also grow to be very large when placed in some sort of agitation. And that can be either a spinning bioreactor, which is shown here in this diagram, but they can also simply be placed on an orbital shaker. And so what we end up with are these really beautiful, quite complex tissues. This is a section through one of these organoids with neural stem cells labeled in red and neurons labeled in green. And it doesn't maybe look too much like a brain in this section, but actually when you look at specific subregions, you can see various regions of the brain. So for example, the retina here, you can see this pigmented region. This is the retinal pigmented epithelium. We also get regions of the forebrain, including the dorsal here shown in green by PAX6, and as well as ventral here shown in red, stained for NKX 2.1. We can also see choroid plexus. This is a region of the brain that normally generates the cerebral spinal fluid. And we can also see other regions of the brain, including midbrain and hindbrain. Here you can see this juxtaposition of the GBX2 positive hindbrain and the OTX1-2 midbrain. And more recently, through work done in Jürgen Knoblig's lab and with a student there, Magdalena Renner, we were able to show this really remarkable self-organizing of these signaling centers, which can actually sort of organize the surrounding brain tissue. So if you look at a brain in vivo, what you find is that in the sort of more central region of the cerebral cortex is a region called the hem, which releases a number of important signaling growth factors, including Wnt and BMPs. And then on the opposite side, you have something called the pallial subpallial border. And these two organizing centers help pattern the intermediate tissue, so the dorsal cortex. And what we found is that organoids, even without any exogenous growth factors, will spontaneously develop these different organizing centers. So you can see on the top there, we've done an in situ for Wnt2b, which will label that hem, and you can see nicely these two sort of hem regions. And these hem regions are between the choroid plexus here in green and the dorsal cortex marked here in red by the TBR2 intermediate progenitors here. So that, I think, was really remarkable. But we could also see these really nice borders of the ventral region marked by GSX2 here in green. This is the bottom image, and the TBR2 positive cells. And you can see this nice sort of juxtaposition of these two tissues next to each other. And this happens completely spontaneously within the organoids. So we're not actually giving them any kind of growth factors or spatial information that causes them to do this. So I think that that's probably the most remarkable thing about these findings. And it turns out that these, so we typically look at these in sections, but if you actually do a 3D reconstruction of it, so this is by using Clarity, what you find is these different brain regions are actually connected with one another. And here we've marked out the ventricular regions, and what you can see is that those ventricular spaces are actually connecting different brain regions with each other. And so there's really some interesting sort of three-dimensional spatial organization happening in these organoids. What exactly can we model with these tissues? So as I said, they very nicely develop the different brain regions. And if we actually zoom in on just the wall of a particular region, so for example, the cerebrocortical region here, what we find is that the cellular architecture is pretty much identical to what you would see in vivo, particularly in the progenitor zones. So what we find is that progenitors line these spaces, these kind of ventricular spaces here, and neurons properly migrate and position themselves in the basal regions of the organoid, just like they would in vivo. We also see that the radioglial neural stem cells display important behaviors that are specific to these types of neural stem cells. So as I mentioned, they have this epithelial morphology. Here I've stained, well, there are a few neural stem cells marked by GFP in an in vivo sample here on the left. And on the right, what you see are in the organoids, you can also nicely see these long basal processes. So they have this epithelial morphology, and they also undergo important behaviors that are specific to these cell types. For example, interkinetic nuclear migration, which is where they move their nuclei in time with the cell cycle, so that they move their nucleus down to the ventricular surface to divide, which you can actually see a dividing cell in this in vivo sample, and then they move their nucleus back up. And so in this movie here, you're seeing one of these nuclei moving along the length of the cell. We also see these other intermediate populations. So I've already shown you a few images where you could see TBR2 stained intermediate progenitors. Here's another nice one showing that these intermediate progenitors are outside the ventricular zone. The ventricular zone's here marked in green by FOXG1. But we also find plenty of these outer radioglia or basal radioglia. These are cells that show the same kind of marker profile, so they express many of the same markers as the inner radioglia, but their cell bodies are sort of displaced from the ventricular zone, and they're displaced by actually, so in vivo they actually there's actually an intermediate region between the OSVZ and the ISVZ, so the outer SVZ and the inner SVZ, and this intermediate region is actually called the inner fiber layer because it shows these fibers, which we actually sometimes see in our organoids as well. In terms of the neurons that are actually made, as I mentioned, the neurons properly migrate outside the ventricular zone. These tissues even generate sort of an intermediate zone, which is marked here by the lower bracket, and then the neurons position themselves on the surface of the organoid, and that's very similar to what you see in vivo. And these neurons are also generated in a timed fashion. And so this is work that, again, Magdalena and I did together, where we looked at different markers of different neuron types. And in vivo, it's known that these different neuron types are generated in a timely fashion. So the first neuron types to be generated are reelin-positive Cajal-Retzius neurons, and that's what you're seeing here. And in early organoids, this is day 42, you see these nice Cajal-Retzius cells on the surface of the organoid. And then if a couple of weeks later, you start to get the other cell populations of other neural layers. So CTIP2 neurons would mark more deep layer neurons, and SAPB2 neurons would mark more upper layer neurons. And these deep layer CTIP2 positive neurons are made a bit earlier, and then the SAPB2 neurons start to be made. And then much later, you start getting astrocytes being made. And this is very similar to what you see in vivo. And actually, we've done some quantification of this and looked at when these various populations are generated. And what we find is this nice sort of transition from reelin neurons to CTIP2 neurons to the SAPB2 neurons and finally to GFAP positive astrocytes. So this shows that not only do we have that kind of spatial patterning that I showed you before, but we also see a temporal patterning of the neurogenesis. In terms of spatial positioning of neurons, there's also some sort of rudimentary layering of these cell types. Now in vivo, what you have are 6 layers of different identity neurons. And while we don't really see 6 clean layers that are sort of well separated and clearly have different identities, what we do see at least are some sort of separation of inner and outer layer neurons. And I think you can see that better in the bottom panel where you have the CTIP2 neurons on the inside and the SAPB2 neurons more on the outside. And I'll come back to that in a little bit. But what we also see are other neuron types. So the cerebral cortex is mostly populated by excitatory pyramidal neurons, but there are also some inhibitory interneurons that migrate into the cerebral cortex from the ventral forebrain regions. So we actually looked at some of these neurons, and here are some calretinin positive neurons labeled, and then you can see the ventral region marked by NKX 2.1. And what you see remarkably is these sort of streams of neurons that are coming from those ventral regions and going into the dorsal. And again, I'll come back to that in just a bit. In terms of neuronal maturation, we've done some imaging of just spontaneous activity. This is a calcium dye. And what we find is that the neurons in these organoids show spontaneous activity where we don't even really have to put anything on them to get this activity, but we can increase their neural activity by putting on things like glutamate, a neurotransmitter, and we can also block it by putting on blockers of synaptic activity like tetrodotoxin. Okay, so I just want to compare a little bit with some of the other 3D organoid, well, neural organoid methods, and just kind of talk about what are some of the differences there and what are some of the strengths and weaknesses of the different methods. So I've already talked pretty extensively about the cerebral organoid method, but in the same year, the lab of Yoshiki Sasai published forebrain organoids, and these are essentially very similar types of tissues, except that they're really sort of driven towards forebrain identity. And they do this by putting on a number of small molecule inhibitors, like a SMAD inhibitor and a Wnt inhibitor, and combining this with HIO2, as well as a few other important sort of components, heparin, matrigel dissolved in the media and such. And they find that they can generate these really quite beautiful forebrain tissues, which you can see here. This is many of which are actually dorsal cortex as well. Then the lab of Sergiu Pasca developed something called cortical spheroids. Again, these are driven towards a particular identity by the use of small molecules. They do SMAD inhibition and then certain growth factors to kind of support the neural stem cells and then to support neurons. And these are primarily cortical in identity and generate these nice sort of rosettes within a spheroid. And just to talk about a little bit sort of what one method versus another sort of involves, I've already mentioned these growth factors in these small molecules, and by putting on these different signaling pathway modulators, you can basically drive the tissue towards a more sort of pure identity. So if we kind of look at this on a spectrum, what you find is that probably the most pure kind of cell population, if you're looking at brain development, would be neural stem cells. Just a pure population of neural stem cells would be very pure and very homogeneous. And on the other end of the spectrum, you'd have whole brain organoids, where you've got all different brain regions, you've got all different types of neural cells in there, and all kind of in various spatial organization relative to one another. And then sort of in between, you've got more or less homogeneous populations, but as you go towards more homogeneous population, you also sort of start to lose some of the complexity. And so what we see is that there's this really interesting balance between the complexity of the tissue that you're generating, where whole brain organoids can give you all these different brain regions, and they will even interact, and you'll even see interneurons going from one region to another, and for example, choroid plexus generating cerebral spinal fluid, which will fill those ventricular spaces and such. But as you go more towards sort of patterning these tissues with growth factors, you get cleaner tissue. So the forebrain organoids, for example, are pretty clean, just forebrain tissue, and that you wouldn't have midbrain and hindbrain in there. But whereas the spheroids are even more clean and sort of more cortical. But as you go, you also find that these tissues are a little bit smaller and have fewer of those different cell populations. So the issue, of course, is as you get the more complexity, the tissues are really beautiful, of course, but they're also less homogeneous. And so there are some issues with that, depending on the particular experiment or research question that one has. Along with this complexity sort of comes the overall size. What we find is that the cerebral organoids generate very large overall tissues, which you can see here in these upper images, but also specifically the cerebral cortical regions themselves are also very, very large. And we think it's probably the interaction of the different brain regions and maybe even the cerebral spinal fluid that's being generated by the choroid plexus. So having these other brain regions, I think, is also important for generating larger and more continuous cortical regions. And I think it kind of makes sense because also in vivo, these different brain regions are not developing in a vacuum. They are developing with other brain regions interacting with them. Right. But actually, in the end, if you just focus on the cortical region, these various methods are very similar. So there are differences in the size of the individual cortical regions, but actually the overall sort of the cytoarchitecture is really quite similar in all these cases. So I'm showing in the upper left is the cerebral organoids, in the middle is the forebrain organoids, and in the bottom right are the cortical spheroids. And what you find is that you have a nice ventricular zone in all cases, neurons that properly migrate out, and intermediate populations in between. So now I'll just talk about some of the new modifications that have been made to specifically the cerebral organoid method. So because it is a beautiful system in terms of the complexity, many labs have started to take up this method, but they're also interested in trying to improve the homogeneity while keeping the complexity. And so that's something that I myself have worked on as well, but also others. So I'll just talk about some of these new modifications. So the lab of Guo Li Ming and Hong Jun Sung recently published a paper where they actually took the cerebral organoid protocol, and there were a couple of important modifications that they made. One is they developed this really cool bioreactor. It's sort of a scaled down bioreactor in terms of each bioreactor is smaller, but scaled up in terms of the number of bioreactors that you can have running. And so you can really scale up the production of organoids. And I think this is a really important step towards being able to do things like drug screening, for example, because scaling up organoids would be necessary for that. They've also been able to describe methods for specifically driving individual brain regions. So by taking the cerebral organoid protocol as a starting point and then using particular growth factors, they're able to drive them more towards forebrain or more towards midbrain or more towards hypothalamus. Now, very recently, the lab of Paula Arlotta was able to generate organoids that would live very long, and using a combination of different factors, but particularly the use of BDNF, they were able to get really great maturation of the neurons within the cerebral organoids, and even some connection between those neurons and neural network type connections between those neurons. And they could even show that in organoids where they had this kind of retinal region, which I described can happen in these organoids, if they shined light on these, some of the neurons would actually respond, and that's shown here on the bottom. So I thought that was really remarkable, that there's this kind of intrinsic network formation, and you can even modulate it with things like light. Very recently, there have actually been 3 studies looking at a fairly similar question, and that is what happens if you actually take organoids, drive them to a particular brain region identity, and then fuse them. And so in Jürgen Knobig's lab, they actually took our cerebral organoid protocol and figured out a method to drive it towards either dorsal identity using cyclopamine or a ventral identity using a Wnt inhibitor and sonic hedgehog agonist. and basically then fused these two organoids together by embedding them together in that matrigel droplet. And once these were fused, they could see that there were these ventral interneurons migrating from the ventral region into the dorsal region, very similar to what you would see in vivo. The lab of Sergiu Pasca also did this, where they showed that they could take spheroids and either ventralize or dorsalize them and fuse them as well, and also show that you have interneuron migration from one to the other. And again, also cortical and ganglionic eminence spheroids, this was recently published in Cell STEMCELL, they were able to show that these could fuse and you could get interneuron migration from one to the other. So this is really remarkable that you can take these two different brain region identities and get this communication between the two brain regions similar to what you would see in vivo. Going back to the sort of reproducibility versus complexity issue, what we'd really like is a system where we could have the complexity that arises intrinsically without any exogenous growth factors, but be able to control it a little bit better and try to get it more reproducibly. And so that's where something that I worked on for much of the end of my postdoc in Jürgen's lab, and then part of the beginning of my own independent position, we worked with essentially a sort of a bioengineering approach where we noticed that the neural tissue always seemed to form on the outside of the embryoid bodies. But embryoid bodies are spheres, which has the lowest surface area to volume ratio. So if you want more surface area tissue, then a sphere is actually the worst shape that you would want to use. So we decided to start using these scaffolds, basically made of little microfilaments, and shape the organoids. We want to keep the self-organizing capacity of these cells, so we only put a few in there. And so most of the cells are not actually touching the scaffold. Most of them are still just touching each other, but it does give the EB this nice elongated shape. And by giving it a nice elongated shape, we end up with a lot more of this neural-induced tissue. And what was really interesting is that just by doing that, we also seem to influence the identity of those tissues. And actually, these bioengineered organoids were almost entirely forebrain in identity. So that was quite, I think, kind of unexpected and very interesting and very useful for us, because most of the work that we are doing is on forebrain. But you can see it's both dorsal and ventral forebrain here. And actually, if you do staining for some of these markers, FOXG1 will mark various forebrain regions, and TBR2 and TBR1 here will mark dorsal cortex. So you can see that these tissues are pretty pure towards forebrain identity. Right, so going back to the layering issue, so I mentioned that in the cerebral organoids that we initially established, we saw a little bit of rudimentary separation of upper and deep layer neurons, but not really like clear layers. Now, in the paper from the Ming lab, they showed that they could actually get these kind of separation of more of these layer markers. Here you can see these different layer markers labeled here, brain2, CTIP2, TBR1, SAPB2, and CUX1. And you can really nicely see how these different layer identities are distributed within this cerebral cortical tissue. So that was really, really remarkable. But what I found really interesting about this whole thing, both in the case of our sort of separation of deep and upper, and the case of their more discrete layering, is that in both cases, you don't seem to get the same kind of structure that you see in vivo. So I think it's really quite interesting how this layering comes about, because what's happening in these organoids is that although these identities are established and generated in a timely manner, when those neurons initially go out into the basal region of the organoid, they look kind of random in organization. They're kind of just sort of randomly dispersed over the surface of the organoid. You don't really see any directionality to the nuclei. Whereas in vivo, you can see that the neurons, when they migrate out and start to populate this cortical plate, they're all radially aligned. You can see their neurons, their nuclei are all sort of elongated and radially aligned, and also very, very dense within this cortical plate. And so I was very interested in how that cortical plate organization comes about, and whether we might be able to model that in organoids to better sort of model the in vivo developmental steps that give rise to a layered cortex. And of course, one of the things that immediately stood out to me is that in vivo you have a basement membrane. You have this very thick membrane of fibroblasts and what will eventually give rise to vascular tissue that really keeps a nice, strong, basal signal for those radioglia that are connected to it. But in our organoids, we don't have that overlying tissue, because that overlying tissue is not generated from within the brain. It's meningeal or the pia. So what I was interested in first was just to look at what happens in organoids. Is there a basement membrane, and what happens to it? And although initially we did find that there is a basement membrane that's established in very early organoids, later on when they start making neurons, this basement membrane essentially breaks apart. And you can see that here stained for laminin. And so we then decided, well, what would happen if we could try to reconstitute the basement membrane by putting in extracellular matrix proteins into the media? And so we did this, and what we found was that the organoid somehow sort of seems to make use of the ECM proteins in the media and maintains the basement membrane very nicely. And the result is really quite striking. What you end up with are this nice radially aligned and dense cortical plates here on the right that you can see. And when you stain for this, these are, in fact, they are neurons here. You can see them marked by TBR1. You can see the double cortin positive processes of these neurons, and the sort of typical lack of double cortin staining within the cortical plate. That's very typical of what you would see in vivo. And we even see this kind of separation of subplate and marginal zone on either sides of the cortical plate, here marked by chondroitin sulfate proteoglycan. Then when we looked at the radioglia, we found that they have nice long basal processes that extend all the way to the outer surface of the organoid. We think this is probably what is giving rise to radially aligned cortical plate. And finally, we're also able to see that the neurons are properly undergoing radial migration. They move with a stereotypical pattern where they sort of move out of the ventricular zone first and into an intermediate zone. And then they kind of spend some time as a multipolar morphology, sort of feeling around, and then eventually they kind of reposition themselves and then move outward. And you can see that in this video here. This sort of large cell is actually an outer radioglia. And then you'll see a number of little neurons migrating out, and you can see them starting to move up and out into the outer region of the organoid. And on the bottom here are traces of that. You can see this, for example, the purple trace, you can see that initially pops out, and then sort of there's some time where it's kind of just fairly immotile, just kind of staying around in this intermediate zone, and then it finally migrates out. So what can we do with brain organoids now? Well, one of the things that we did right away was to see, well, what happens if we make organoids from another species? Since we're interested in some of these features that are unique to humans, we wanted to look and see, well, what does a mouse organoid look like? And so it turns out that actually, quite remarkably, the mouse organoids are overall much smaller than human. And in particular, the individual cortical regions here are much, much smaller. You have much less continuous kinds of lobes here. And then, of course, we wanted to do, well, if this is truly a model of human brain development, then we should be able to model certain human disorders. So we actually showed in our initial study that we could model a disorder called microcephaly. Now, many of you may be aware of this because it's a disorder that's associated with the recent Zika virus outbreak, and I'm going to talk about that in a moment. But we actually modeled a patient who had a genetic form of microcephaly. And you can see the MRI from this patient. The brain is overall much smaller and has a more simplified gyral pattern. And when we took skin cells from this patient and then reprogrammed them to iPS cells and then generated organoids, could very nicely see that the organoids were much smaller and had fewer neurons and fewer neural stem cells. And what was really great about this is that we could, because it's an in vitro system, we could kind of go back and try to see, well, what was the defect that gave rise to these overall smaller organoids? And what we found is that the neural stem cells early on actually seem to undergo premature neural differentiation, which then sort of exhausts the neural stem cell population, and that results in eventually overall fewer neurons. Now, more recently, there was in work from Flora Vaccarino's lab, they were able to make, now these are 4 brain organoids according to, or very similar to the Society protocol that I mentioned. And they were able to generate these 4 brain organoids from patients with idiopathic autism. And this is just one of the examples here, but what they found is that in their patient cases, there were overall more interneuron type cells in the organoids than the controls. And so this suggests sort of an imbalance between the excitatory and the inhibitory neural populations, which has been implicated potentially in autism. It was very nice to see this type of phenotype in the organoids generated from those patients. Now, I mentioned Zika. So one of the really clear avenues to go with brain organoids is to see whether Zika virus had an effect on brain organoids and whether we could find out which cell population was affected. And so a number of studies have actually done this. I'm just showing one example because it was actually also done in the Ming lab in those same study that described the SPIN-Omega bioreactor system. And they showed that when they put Zika on their brain organoids, they were overall much smaller, and it was very toxic to the neural stem cells. And so by essentially killing the neural stem cells, you're inhibiting the ability of them to make large numbers of neurons. And then very recently in the Pasca lab, this was again, this is actually still the study looking at fused spheroids from both the dorsal and ventral region. What they were able to do is look at patient-derived fused spheroids from a disorder called Timothy syndrome, which is a pretty severe epilepsy, and there's some problems with interneuron migration. What they find is that they can very nicely see these kind of problems with the interneuron migration dynamics. So basically, this is just to kind of give an overview of what cerebral organoids have given us so far. As I mentioned, we've already used the original method for a number of studies, looked at comparisons of human and mouse, human and microcephaly, and other studies have looked at Zika virus with the original method. But now more recently, there's these nice SPIN-Omega bioreactors that have been described and have also been used for Zika virus studies. Now there's been a number of these sort of more directed differentiation protocols described to generate specific brain subregions just by using a few little growth factors. So I'm just showing as an example the example of the dorsal versus ventral regions developed in Jürgen's lab. And then we also described a modification of the cerebral organoid method in using these microscaffold bioengineering approach. We call them engineered cerebral organoids, or ENCOREs. And we can very nicely generate more reproducible forebrain tissue that later, with the combination of dissolved ECM components, generates a nice radialized and condensed cortical plate. So what are some of the future directions? Well, I think it's very broad possibilities here, and I hope that there will continue to be a number of methodological improvements and also modeling of various diseases. We're already seeing that they're being used to model a number of diseases, but I think with further improvements to the method, we'll see even more. And I mean, I think the possibilities are really endless. Essentially, any neurological disorder, I imagine we will start to see people modeling those in this system. And then in the more sort of long-term future, I hope we can start seeing potentially drug screening. So we can generate a cerebral organoid model for a particular disease, and then of course making sure that we use highly reproducible methods. Then I think there's a lot of potential for drug screening as well, and identifying specific drugs that may work for that disorder. And some of this could even be personalized to a particular patient. Now, this is already being done to some degree in other organoid systems like intestinal organoids, where you can actually take organoids from a patient, for example, a patient with cystic fibrosis, generate a number of these organoids, and then try the various drugs that are on the market currently for cystic fibrosis and see which ones are most likely going to work for that patient. And then you can actually go back to that patient and say, look, probably this drug combination is going to work best. So I think it would be quite powerful to use brain organoids in that fashion as well. And in the even further long-term future, I think there's also potential for cell replacement therapies. Now, obviously, we're not going to replace a person's brain with a brain organoid because it wouldn't come with all of the memories and such that make you who you are. But there are specific brain regions that I think we could try to replace that wouldn't change you as a person. So for example, in the case of spinal cord injury, for example, or potentially substantia nigra in the case of neurodegenerative disease, I think that there are possible regions that we could try to continue with and try to see if there's ways we could look at cell replacement therapies. But I think this is much further in the future. In the meantime, I think there are a number of improvements that will need to be addressed when it comes to cerebral organoids. I didn't talk too much about it, but these organoids, of course, they're in a dish. They don't have any of the supportive fetal tissue that would normally be present, and that includes vascularization. So that means that the very center of the organoids is essentially dead necrotic tissue. And so although we can look at the outer regions, so each of those sort of lobes that sticks out of the organoid is very nicely organized. But each of those regions, they can connect to some degree, but everything in the very center is going to be dead. And that's a problem if you really want to look at connectivity between different regions and how they sort of interact with one another. And then later, each of those lobes is still limited in terms of how thick it can get. So you can get a little bit of layering, for example, but you wouldn't be able to get the entire very large, thick, 6-layered cortex because that thickness is simply too large for nutrients to passively diffuse into. So I think that's going to be an important development that we will need. I also think that patterning these organoids, more than just sort of patterning the whole organoid towards a particular identity, but patterning them in a way that enables the development of various brain regions, but in a specific spatial configuration. I think that's going to be very important. And that brings me also to this sort of even better reproducibility, because ideally we would get this complexity and reproducibility all in one, because yes, the human brain is very complex, but actually in an embryo, the human brain always develops with the same shape and overall spatial organization. So there should be a way that we could control this a little bit better in vitro as well. And finally, I just want to leave you with one important take-home message. And I think at the moment, with the current state of the art and the methods that are out there, if you're considering which method to use, it's important to consider this sort of trade-off between homogeneity and complexity. And if the question at hand needs this important sort of tissue morphology and morphogenesis, then it may be best to go down the route of using these complex whole brain organoids. But if the question is specific to a particular brain region or specific to a particular cell type, you could potentially go for a more homogeneous model system like spheroids or rosettes. And so with that, I'd just like to say thank you. I hope that I managed to convey how much I enjoyed my time in Jürgen's lab and how vital he was for this project, his input and his guidance for the development of the organoids in the first place, and then for the improvements that we've made since. As you can see, his lab is continuing to make important strides in this area, and I actually look very much forward to seeing what others in his lab will accomplish. I also want to thank in particular a couple of members of his team, Simona and Hillary, for help, especially with the bioengineering project that I talked about, and then of course members of my lab for their general motivation and excitement about the project and making it a wonderful place to work here. And thank you for listening.
Thank you very much, Madeline. That concludes our presentation. I really want to thank everyone for attending, and I'd especially like to thank you, Madeline, for your time and an excellent presentation. Thanks again, and goodbye.